The African Baobab tree is the national symbol of Senegal. It is considered spiritual in some southern and western African cultures. The tree is an excellent source of vitamins and minerals, possesses many medicinal properties, and provides shade for rural communities. The baobab tree can live for thousands of years, supplying natural and cultural resources for African communities for generations. This is why it's called the Tree of Life. It is imperative that we protect this sacred tree, that in turn protects and sustains human life. ARS geneticist Hamadou Sanaku grew up in eastern Senegal, where he watched his parents and older siblings use the tree's bark, leaves, and fruit for a variety of purposes.
The bark can be used to make ropes, and the pulp from the fruit is used to make drinks. Also, both the pulp and the leaves are included in many food recipes across West Africa.
The once prominent African baobab tree is now being threatened by climate change, human activities such as land clearing, wildfires, and drought. If the tree of life were to further diminish or even become extinct, communities would lose an essential fabric of their lives. In 2018, Hamadou went to Senegal and collected seeds of the African baobab tree. Very little work has been done on these trees to understand their genetics. He wanted to gather basic genetic information, hoping that maybe someday that genetic information can be incorporated into a program to protect the tree of life. Sakanuko collaborated with U.S. Forest Service geneticist Dana Nelson and Nurul Faridi to determine the genome size and the number of chromosomes in the African baobab tree.
Culturally, economically, and spiritually, the baobab is an important tree in Senegal and also in many other African countries. And yet, there is very little research done on it. And that is the reason why we decided to conduct some basic work on its genetics, starting with determining its genome size, chromosome number, and also characterizing its DNA organization.
This proved challenging as this particular species, Adoncinea digitata, had leaves that were rich in complex sugars, making genome size determination and analysis difficult. This species also had a high number of chromosomes, and the chromosomes themselves were quite small. However, not to be deterred, the research team applied innovative scientific methods to create a new chromosome counting technique that could be used for the entire baobab Adoncinea genus, as well as other plants with high chromosome counts. This can be useful for agriculture, and in particular for ARS and Forest Service horticultural and forestry programs that are often dealing with plant species with unknown chromosome numbers. But these basic genetic findings can also pave the way for a more efficient and effective genetic conservation and domestication of the baobab tree.
My hope is that scientists, particularly those in Senegal and other African countries, conduct more research on this very important baobab tree species so that it can be not only conserved but also improved for future generations to come.